Cladograms by Emma Ficker. Cladograms are part of the science of taxonomy. Taxonomy is the science of classifying living things, and this is exactly what a cladogram does. Before we can learn the specifics of a cladogram, it is important to understand the hierarchy of biological classification. This is the way that scientists have used for many years to help organize and classify the 2 million different living things found here on Earth. These topics have helped develop cladograms. The topics are domain, kingdom, phylum, class, order, family, genus, and species. Now, what exactly is a cladogram? A cladogram is simply a diagram that is used to represent a relationship between groups of animals. It is used to visualize the groups of organisms being compared, how they are related, and their most common ancestors. Cladograms contain parts that help organize the organisms being compared into a way that is easy to understand. These parts would include a root, and this is the initial ancestor common to all organisms within the whole cladogram, branches, which is given when a new trait arises that sets apart those organisms, internal nodes, where branches split and represent hypothetical common ancestors, and terminal nodes. These occur at the ends of lines and represent the organism and their relationship. A cladogram takes the characteristics that are shared by an organism and then puts them together by a hypothesis of how you think that they are related. In this cladogram, every animal after the hagfish has jaws, every animal after the perch has lungs, every animal after the salamander has claws or nails like the lizard. At the pigeon, since it is the only being on the cladogram that has feathers, yet still has a jaw, lungs, and claws or nails, it is given a subgroup of feathers. Then after that, the animals that follow, the mouse and the chimp, have fur and mammary glands, and everything else is to before, except for the feathers, because it is not included in the subgroup. My article is titled Cladograms with Path to Event, or CLAPTE, a novel algorithm to detect associations between genotypes and phenotypes using phylogenies. Included in my article were 2 case studies that showed cladograms with path to event. This means that cladograms would not only show the ancestry of certain things, but also reactions that would occur in certain events. They decided to take on the study to prove that CLAPTE is a better way for scientists to test bacteria and their effect on different genes, and explain that it is a more efficient way to find and organize information. CLAPTE tests for character pairs close together on the cladogram. CLAPTE was compared to 3 existing methods: independent contrast, mixed models, and likelihood ratios, in order to detect character pair associations that changed. The CLAPTE, as well as other methods being compared, showed the ancestry of HIV ENV, HIV promoter, and bacterial DNAJ and GUAB. The scientists used CLAPTE and took these results and organized how they would react in resistance for influenza and for associations between different factors. These factors would include the environmental niche, the host organism, the host tissue, the predator or prey relationships, drug exposures, the presence or malfunction of an individual gene, protein, or network interaction, and substitutions, insertions, or deletions in individual nucleotides or amino acid positions. The data included showed both true positive associated character pairs and true negative non-associated character pairs. This was then used to assess errors. Here is what CLAPTE looks like. As you can see, it has many branches that follow ancestry, but you can also see that it branches out based on how it reacted with certain factors involved. In conclusion, CLAPTE is able to show a wider range of information rather than just a select few. The CLAPTE is more efficient in the event where there are multiple factors from one descent, and it also showed to be easier to understand and gain the knowledge you need about certain organisms. Thank you for watching.